Hello, welcome to the introduction of Sanyou Biopharmaceuticals, and thank you for your interest in becoming a partner of Sanyou. My name is Ren Yan, and I am the Technology and Project Director of Sanyou. In this presentation, I'm going to show you why Sanyou is a trustworthy partner for your therapeutic antibody discovery and development. Next, I'm going to show the overview of Sanyou. First of all, Sanyou is a CRO company providing integrated solutions for therapeutic antibody discovery and development. From our founding, Sanyou has always been focusing on the high-quality, high-throughput integrated R&D and value transformation of antibody drugs. Counting to today, there are over 150 employees in Sanyou, and more than 50% hold a PhD or master's degree. Now Sanyou has more than 8,000 square meters laboratory, containing 10 functional modules, which is comprised of over 40 technology platforms. The feature technology of Sanyou is a series of super screening phage display antibody library. To fulfill our mission, we have established 10 functional modules and 41 platforms covering the whole R&D process from the selection of our target to the IND approval. The 10 function modules have more than 120 employees, and they exert the R&D function, including antigen preparations, antibody library construction and screening, in vitro and in vivo efficacy tests, antibody engineering, antibody production and QC, production cell line construction, lab-scale process development, pilot-scale production, and QC. Sanyou is featured by a series of filling-level libraries. The first one I'm going to introduce is the sub-filling human recombinant antibody library. As of 2019, 45.6% approved antibody drugs are fully human. Among the above, 25.8% are discovered by phage library. This is why we believe that the phage display technology is a promising method for the antibody discovery. From 2018, we started providing services with our sub-trillion fully human library. This library has a leading capacity and averagely yield dozens to hundreds binders. We have analyzed the yield of 19 projects, and the average yield is 100 unique binders, and the maximum yield is 3,070 unique binders. We also analyzed the germline distribution of the approved antibody drugs, and we designed our library accordingly, so they have similar germline distribution. The secondary library I'm going to introduce is the immunization library. The immunization library is an advanced technology that combines the animal immunization and phage display. Comparing to the conventional hybridoma technology, immunization library could save 3 months' time. Acquire antibody sequence at the early stage and generate species and epitope-specific antibodies. The immunization library averagely yield about 40 binders, and they usually have nanomolar level affinity. The third library I'm going to introduce is the combined subunit single-domain antibody library. So far, the single-domain antibody library includes alpha immunization library, which has a capacity of 5 times 10 to the 8th. Alpha naive library, which has a capacity of 3 times 10 to the 10th. And a semi-synthetic library, which has a capacity of 10 to the 11th. Single-domain antibody is an emerging form of antibody drugs, especially for the generation of bispecific and multi-specific antibodies. The next featured services are our one-step solution for antibody engineering. With our conformational analysis and design, we can deliver humanized antibody with over 95% humanized and non-reduced affinity or function. We provide integrated services for humanization design, antibody expression, and validation. So far, we have completed more than 100 humanization projects. Over 40 antibodies are in the IND filing stage, and 7 antibodies are being tested in the clinical trials. We also provided affinity maturation services with constructing libraries of 1 billion capacity. This method can improve the candidate antibody's affinity, alter its pH sensitivity, or improve its stability. We have also delivered more than 100 affinity maturation projects. Sanyou also provides a 14-day protein preparation service. We use 2 transit expressing systems, including the true and the hacker 293 expression system. The yield is as high as 500 milligrams per liter, and we can produce more than 200 proteins per week. The protein qualities are verified by SDSPH, SEC, LAL, and ELISA for the activity. For as low as $200 US, you can get milligram level of high quality proteins. For production, cell line construction, still provides services with 3 sites of replicated GMP cell lines. We can deliver single clone stable cell lines within 3.5 months and finish batch scale process development within 3 to 4 months. The antibody yield for monoclonal antibody is as high as 8 grams per liter, and for bispecific antibody is as high as 6 grams per liter. Sanyou also has pre-incubated antibody drug projects which are ready for licensing. The first project is the COVID-19 neutralizing antibody. This fully human antibody has outstanding affinity and blocking activity. It also has strong efficacy against pseudovirus and live virus in vitro and in vivo. This antibody received the IND approval last year. Sanyou sincerely invites you to join our partnership. Together we can serve more and achieve more. Thanks for your time. We look forward to talk to you soon.